Cheese manufacturing process. Cheese is a concentrate of milk fat and milk proteins. Cheese is a highly proteinaceous food which is obtained from the milks of animals such as sheep, goats, buffalo, the llama, yak, reindeer, ass, or cows. About the thousand types of cheeses have been described depending on the properties and treatment of the milk, the method of production, conditions such as temperature, and the properties of coagulum, and the local preferences. Steps involved in the making of cheese. First step is standardization of milk. The quality of milk has effect on the nature of cheese. Cheese made from skim milk is hard and leathery. The more fat a cheese contains, the smoother it feels to the taste. The fat as to protein ratio is often adjusted through fat addition in order to yield a cheese of consistent quality. After the collection of milk, it is clarified by centrifugation and stored at low temperature to prevent bacterial growth. Pasteurization may be preferred, which is a high temperature short time treatment or long temperature short time treatment. However, the organisms present in raw milk are also important during the ripening process or maturation of cheese process. The milk may also be homogenized by forcing it at high speed through small orifice to reduce the milk fat globules for use in production of soft cheeses. Second step is inoculation of starter cultures. Starter culture bacteria utilized in this process are lactic acid bacteria, which are responsible for the production of lactic acid at a desirable temperature of 40 degrees Celsius. This temperature may vary and it is dependent on the type of lactic acid bacteria used. For example, 40 degrees Celsius temperature is better for Lactococcus lactis, whereas a little higher temperature is favorable for the thermophilic lactic acid bacteria such as Streptococcus thermophilus, Lactobacillus bulgaricus, and Lactobacillus helveticus. Lactic acid bacteria in this cheese making process has the following effects. First, it causes the coagulation of casein at pH 4.6 due to production of lactic acid. It provides a favorable low pH for the action of rennin, which is a proteolytic enzyme, the enzyme which forms the curd from casein. The low pH eliminates proteolytic and other undesirable bacteria. Shrinkage of curd and thus promotes the drainage of whey. Lactic acid bacteria produce many metabolic products such as ketonic compounds, esters, and aldehydes, which are important for the development of flavor in finished cheese during the maturation process. Next step is addition of rennet. Rennet is responsible for the formation of coagulum. In industrial process, the initially the milk is inoculated with lactic acid bacteria. It is allowed to be incubated for 30 to 40 minutes for the activity, bacterial activity, and later on it is inoculated with this preparation called as rennet or rennin extract. Rennin is derived from the fourth stomach, abdomen, or well of freshly slaughtered milk fed calves. Besides those of cows, the uh, abomasum of kids, that is, young goats, lamb, or other young mammals, have been used. So, this preparation is used as a source of rennin. Rennin is a proteolytic enzyme which is responsible for the coagulation of milk, which is responsible for the digestion, partial digestion of casein. Rennin is also called as chymosin. There are other sources of this proteolytic enzyme, that is rennin. That is, it is obtained from different types of microbes. First, the pH of milk is brought down from 6.8 to pH 5.5 by the action of inoculation of lactic acid bacteria, which produce lactic acid from lactose in milk. Following are the examples of different types of proteolytic enzymes or rennins, which are obtained from different microorganisms. For example, harmylase is a type of proteolytic enzyme obtained from Mucor mici. Mucor mici seems to be a very commercially important organism for as a source of these different types of rennins. The effect of rennin. Rennin is responsible for catalysis of hydrolysis of kappa casein to release para kappa casein and kappa casein macropeptide. Casein consists of three units: alpha casein, beta casein, and kappa casein. It is a hydrophobic protein, and kappa casein is responsible for keeping the micelle of whole casein molecule soluble in the aqueous milk. So kappa casein is responsible for maintaining the solubility of micelle in milk solution. If this kappa casein is broken down, the hydrophobic sites of this casein are exposed, and which are responsible for the formation of coagulum. So, this enzyme that brings the hydrolysis of kappa casein, it results into formation of para kappa casein and kappa casein macropeptide. Out of these two, para kappa casein is strongly hydrophobic, and this binds together with other molecules to form a solid curd. The events up this coagulation are aided by lowered pH and by increasing temperature up to 45 degrees Celsius. Most of the bacteria, fat, and other particulate matter are entrapped in curd. When casein is removed, the remaining liquid, it contains protein, that is lactobulimine, globulin, and yellow-green riboflavin, that is vitamin B2, which is left in whey. Next step is shrinkage of curd, that is the water content of the curd is decreased by the method of heating, or it is followed by cutting the curd into smaller pieces and by means of application of pressure in order to in order to decrease the water content of curd. Next step is salting of curd and pressing of curd in order to prepare the blocks of particular shape. Salt is important not only for the taste, but it also contributes to moisture and acidity control. Most importantly, however, it helps to limit the growth of proteolytic bacteria, which are undesirable. The curd is pressed into shape before being allowed to mature. There are two methods of salting. First is by dipping in brine. Brine is a initial solution, that is a salt solution. So blocks are kept dipped. Blocks of curd are kept dipped in brine for a particular period of time. And the another method is by feeding solid curd into mill for the formation of small pieces, and then dried salt powder is sprinkled with continuous mixing. These are the two methods of salting. Next step is the cheese maturation or cheese ripening. It is a slow joint microbiological and biochemical process which converts the brittle white curd or raw cheese to a final full flavored cheese. So in order to develop a particular taste and flavor, cheese needs maturation process. For this, the responsible agents are the enzymes which are present in milk, enzymes present in rennet, that means rennins, proteolytic enzymes, and the enzymes which are released from the starter culture microbes, that is lactic acid bacteria, as well as the microbes that develops during the process of maturation in cheese. Among the cheese whose particular characteristics are dependent on particular organisms are blue veined cheese or Roquefort cheese. Other examples are also listed. So this blue vein cheese, as name indicates, it contains blue patches due to the growth of a fungus called as Penicillium roqueforti, which is responsible for the maturation of this cheese. Another example is Swiss cheese, with its characteristic flavor and holes produced by fermentation products and gases from Propionibacterium species. Yeast, Micrococa, and Brevibacterium linens impart the characteristic flavor to Limburger cheese. One of the example of soft cheeses, that is camembert, the protein is completely broken down almost to amino acids, whereas in the hard cheeses the protein remains intact.